Welcome everybody to the webinar here today, Fulfilling the Promise of Ag Biologicals, focused on the topic of delivery. My name is Jane Fife. I'll be serving as moderator today. I'm Chief Technical Officer at 3Bar Biologics, the sponsor for today's webinar. And I'd like to mention that this is the second webinar in the series that we're calling Fulfilling the Promise of Ag Biologicals. The first webinar we actually held last fall, and it was on the topic of shelf life. So you can find the recording of that webinar on our website at www.3barbiologics.com. We're going to be recording today's webinar as well, and it will be available on our website afterwards. We're leaving time towards the end of the panel discussion for live Q&A. So please continue to introduce yourselves in the chat box, those that are joining as participants, but also add questions at any time during the webinar, and then we'll work to answer those questions after some of our panel discussion. So let's get started. Joining me today are four distinguished panelists, each coming from different areas of the ag industry and each having different experiences and perspectives on delivery of biological products. Our panelists include David Flack, VP of Business Development at New Leaf Symbiotics, Dan Poston, Director of Agronomy at Pivot Bio, Adam Kayser, Bioag Agronomist at Novozymes, and Jim Lappin, Director of SimPass Product Portfolio at AMVAC. And I'd like to personally thank each of you for joining us today and taking time out of your day to be part of this panel discussion. I'll have each of you introduce yourselves here momentarily, but first I kind of wanted to set the stage. Today's topic deals with delivery of microbial products. And so dealing with living organisms being delivered into the field, obviously the importance of the viability of those organisms through the delivery process is important. And a lot of times there's focus on early in R&D on fermentation, formulation, and while that's really important, I would argue equally important is how we're actually reducing this to practice and delivering these products in the field. And application technology, what's available to us today was developed for synthetic chemistry. And so a good question to ask is, are these technologies good enough for living microbes, or do we need to think differently? And so with that, I'd like to have each of our panelists introduce themselves and also maybe talk a little bit about comment on delivery practices and any issues that they may have encountered in their own practice. And so David, if you'd like to start.
Sure, thanks, Jane. Yeah, my name's David Flack. My background, I've got about 35 years experience, supply side agriculture. The first half of that, synthetic chemistry, predominantly with Syngenta and Scott's Miracle-Gro. And then the last half of that, up to today, has been in the biological space with smaller privately held companies, private equity ventures like New Leaf Symbiotics. My actual biological experience, I've worked with dry and liquid products and row crops, predominantly wheat, soybeans, corn, applying them in furrow. New Leaf uses a practice to apply at the planter box in combination with seed lubricants, which is a new and developing way to get biologicals to the furrow. And then foliar, of course. And then in the specialty markets that I focus on for New Leaf, where this all actually gets a little bit easier, we use drench and irrigation systems to get our microbes where we need them to be in specialty production, indoor and outdoor. It doesn't remove the challenges. We still have to deal with compatibility and shelf life and those kind of things, all solvable, but certainly considerations when you're working with a biological versus a synthetic.
Great, thanks, David. Dan, if you'd like to go ahead and introduce yourself.
Yeah, thanks, Jane. My name's Dan Poston. I am the Director of Agronomy for Pivot Bio. But in reality, I'm really at the interface between agronomy and product development. So most of my career has been in the crop protection sector, really trained as a weed scientist. Spent the last 15, 20 years making myself a pretty decent agronomist. My passion really lies in developing cropping systems and figuring out how all of the pieces come together to produce a system, how we incorporate all these pieces on farm at the grower interface. So I've really existed professionally at the point of handoff to the field and what happens when products really hit the farmer's field. So that's kind of where I fit into the picture. From an application standpoint, I've kind of worked with most everything at the applied level. Early on, everything from biopesticides to inoculates to other biologicals in the professional sector. I spent about a decade working in the university system, and then another decade working for a major seed company before going to work for Pivot. So I've kind of seen it all. Most of my professional career in the South. So from a biological standpoint, we have low organic matter soils in the South, high temperatures. If there's a chance to kill a biological, we can do it in the South. So I've got a lot of experience trying to see pieces and parts of things that haven't worked and things that have probably led to a lot of the past inconsistencies in biologicals. So lots of experience there. I'll just stop there, Jane, and we can take questions or whatever as we go on.
Sure, thanks, Dan. Adam, I think you're next.
Good morning or afternoon for everyone, depending on where you're at. My name is Adam Kayser. I live in Wells, Minnesota, so southern Minnesota. I've been working specifically with biologicals in agriculture for just a little over 15 years, and I got my start working primarily with legume inoculants. And what's really neat, I guess, you know, as this industry has evolved, legume inoculants has been, you know, the longest standing biological technology we've had in the United States. You know, we've been using these technologies for 120 years, and growers used to apply it, you know, on the farm just before they seeded alfalfa and clover. I've worked with, you know, when you get across the border and into the pulse growing regions of the United States, we get a lot of granular products because they have the equipment capabilities with those big air carts to move a product like that. Of course, seed treatments from liquid to dry, planter box, like David had mentioned. In-furrow technologies is certainly where I've seen the most advancement here in the last few years. And then also foliar applied technologies, and those are primarily on would be the biocontrol sector.
Yep. Thanks, Adam. And Jim, last but not least.
My name's Jim Lappin. I work for AMVAC. AMVAC is a kind of a mid-range company with a broad portfolio of products. My own experience, I have over 40 years in ag chem, ranging from large multinational. I've been in both domestic and global roles, predominantly sales and marketing. At AMVAC now today, biologicals are a growing portfolio between our row crop, specialty crop, nursery supply, consumer goods business. We have over 80 SKUs of biological products that we're currently selling, and that would run the gamut of everything that's been already mentioned, from a in-furrow seed treatment to foliar applications, both in conjunction with synthetics or in place of synthetics in some cases. So again, AMVAC sees biologicals as a growing category of products and a growth platform. We've recently launched the SIMPAS application system, which we believe will be a unique way to deliver biologicals to a broad range of crops in the US and other countries.
Great, thanks, Jim. All right, great introductions and a lot of different ways to apply biologicals were discussed. We're gonna, in this first segment, delve a little deeper into, you know, reducing this to practice, how the microbes are being delivered, including equipment, and hopefully you can comment further on what's worked, what hasn't worked based on your experience. But Dan, let's start with you and talk about your experiences. I know a lot lately has been with in-furrow delivery of Pivot's proven product in the field. Maybe that's a good place to start.
Jane, what specifically in that arena are you looking for? Are you just
Just as a starting place of this is one way we can deliver in furrow, and what's your experience have been, and maybe reducing it to practice with farmers as a starting place, and if it's been successful in what you've seen in the field.
Well, as you enter the in-furrow market, which we're playing in right now at Pivot for sure, the first thing you've got to look at is what folks are actually doing in the real world. And most of those guys they're mixing everything under the sun, number one, in the trench. That's everything from fertilizers to biostimulants to carbon sources to humic sources. And so the first thing you've got to figure out is how your biological plays in that space. So are these biologicals compatible both physically and biologically to fit into that trench? And so you've got to do your homework on the front end to make sure that once you put that microbe in the trench, that you can deliver a live microbe to the trench. So that is in my mind, not only with our microbes but others, that's one of the hurdles in the industry we've got to overcome, and that's are these things truly biologically compatible with the application technology we're using? So that to me has been the biggest hurdle. We've been quite successful in that space. We spend a lot of time with our customers really screening their products that they're mixing in trench to determine if that's a successful marriage with our microbes. So that we spend a lot of time there, Jane, to be honest with you, and we've been quite successful there, providing that service to our customers and getting our microbes successfully entrenched with farmers.
Right, great. Adam, I'm going to have you follow up here. I know you've had a lot of experience with both in furrow and those compatibility issues and seed treatment as well. What are your thoughts here on how we're approaching reducing this to practice and what's being delivered in the field?
Dan hit the nail on the head, understanding what growers are doing today in their specific regions, what equipment is available to meet their needs and to meet the needs of the microbe. I guess I'll comment on seed treatment a little bit more. Applying biologicals to seed is probably a little more understood and practiced, when you consider, most of the soybeans, come to your dealer untreated and then ultimately get treated with a fungicide, insecticide, and likely a biological. So the same practices that we've learned in that space, I think, is translated into in furrow, trying to keep the organisms separate for a compatibility standpoint, so they're not taking a bath in some of these chemistries and nutrients. Other things that have translated, I think, well is, one thing that always makes me nervous are some fertility products, specifically heavy metals. There's a lot of offerings out there in the marketplace with things like boron and zinc and copper, and both seed treatment and in furrow are great ways to deliver those solutions, but we need to be mindful that they might not always play nice with the other things we wish to put in the tank. So keeping those things separate and understanding, and that's, I think everybody in the industry has done a really good job, because ultimately, it's difficult to assess, when products are working and not working when it comes to biologicals. That's again where I think it's a really good starting point to understand legume nodulation and nitrogen fixation. That's a very visual biological. If it worked or not in conditions where if you didn't have success will not show. So yeah, and things like this, get-togethers like this and transferring this knowledge to the broader arena, I think, is key in success of biologicals. I think stewardship is one of the biggest hurdles the industry has.
Oh, that's great. We'll circle back around to indicators maybe later, but the fact of inoculants being a good indicator, right, with that root nodulation is a good point. Let's see, David, I know you've had experience working with specialty crops, as you mentioned, And maybe any different considerations or other considerations based on your experience with row crops too.
Well, I think one of the key things, at least, and I'll keep my comments kind of towards the greenhouse here, but it plays the same as it does in row crops. You've got to arrest these microbes' metabolism to get shelf life and stability. And for us, water activates our microbes' metabolism. So we've had to do a lot of work, a lot of research. It's taken several years and a fair amount of investment, to figure out how to do that, how to arrest the microbes' metabolism. And so we're a spray dried powder now. We've got about a year shelf life, and that's a key thing in terms of the grower being able to handle and manage inventory. Even longer is better, but when we first started, it was only 3 months. So we've come a long way. So that's one of the key points. Numbers is another key point. We want to end up with about 100,000 cells per plant when we're treating in the greenhouse. So we have to build highly concentrated formulations coming out of fermentation. Of course, keep that viable, but get it to the grower in a way that he's not handling a lot of product at the same time, and making it easy to dose it into that water on a per unit basis and get that 100,000 that we're looking for. So we spent a lot of work really on the formulation product development side to get where we need to be for the grower. And then, like I said earlier, it gets a little easier once we get to the greenhouse, because we want to go in water, a drench or their irrigation system is easily available, and it makes for a really great way to treat in a greenhouse environment.
Yeah, great for that perspective. And Jim, I don't know if you had anything else to add here.
Well, it's interesting when we have this conversation about the equipment that are used to apply products at plant or seed treatment or even foliar. It ranges based on the region. So just to give you some examples, when we think about planter systems in the marketplace, only about 1/3 of planters in the market today have at plant either 2x2 or in-furrow fertilizer capability. So you have a set of growers, maybe regionally disproportionate in the upper Midwest, that are used to applying products in furrow with fertilizer. But as those mixes become more complex, as they add, most people aren't selling single micronutrient blends, they're selling combination micronutrient blends. Maybe there's a biological in there, maybe there isn't. You got fertilizer variability. If you were to go survey 10 different suppliers of 1034O and look at those formulations, you'd find marked difference between how well those 1034Os stay in suspension. And then you get into the biological component to say, well, the biological product I'm going to add into that system, is there a lot of suspended solids? Will it settle out over time? So when we talk about things like shelf life, there's shelf life in the jug, there's shelf life in the supply chain, and then there's shelf life in the furrow relative to all the things it's interacting with. And when you think about seed treatment as another category, where is the seed being treated? Is it treated upstream in the bag? Is it treated at the retail plant? Is it treated on the farm? That all has an impact on your viability of your material that you want to put in furrow. And then you also have to ask the question in terms of what's the expectation of that dose rate to get to the biological effect that you want. As we talk to seed treaters, they're putting lots of things on that seed. So at some point you just run into physical limitations of how much physical product can you put on the coat of that seed without creating a problem of flow of the seed or storage in the bag or whatever the case may be. So again, there's numerous challenges for the various ways we apply seed treatment, fertilizer. Foliar perhaps has a few less challenges, but then it's most cases they want to be applied with another pass, and that's almost always going to involve a synthetic chemistry, and some of those synthetic chemistries have bactericides they put in the jug. So again, you just have to be aware of all the components that you can potentially be mixed with, be in conjunction with, and think through the viability of your microbes.
Yeah, and I'd like to build kind of on that thread. And Jim, I'm going to have you actually start this next round of addressing. You guys have talked around a lot of the different delivery technologies and so forth out there. and whether or not biologicals fit well in terms of whether it's a seed treatment and furrow into a lot of liquid. Let's this go around, think about it in terms of if we were to reduce this to practice in the best way for the biological, knowing that the farmers are doing a lot of different things, what makes the most sense in terms of what we're applying with and liquid versus dry. You know, we could bring in formulation, but if we were going to improve what we're doing with the microbes, what makes the most sense in terms of what the farmers are already doing, reducing it to practice? And maybe Jim, starting with you, I know, you know, with AMVAC SIMPAS system, there is some rethinking going on here and how we're delivering, and maybe, you know, talk about fitting in with grower practices with some of these new ideas in mind. If you wouldn't mind starting, that'd be great.
Sure. So with SIMPAS, and just for the audience, SIMPAS is an aftermarket system that can bolt on to most planters, and it delivers up to 3 products, can deliver either a linear down the row, can deliver variable rate, or can deliver just within precision management zones. So again, it's capable of delivering 3 products per row prescriptively to full rate in-row to in-row. So growers can apply the product where they think it has the most biological effectiveness, where they are targeting a specific pest problem, or there are a certain set of conditions where a biological or a synthetic is going to perform at a higher level because of those conditions. So SIMPAS is kind of a reimagining of in-furrow, where traditionally we've had granular insecticide boxes, we've had in-furrow systems and seed treatments. This kind of steps away from those challenges, as if I can't put enough of my dose of a biological on as a seed treatment, running it through a SIMPAS system, I may still have a dose in my seed treatment, but I can complement or supplement that by an additional dose through the SIMPAS system. I may have fertilizer that is mostly compatible, but not always compatible. By separating those in SIMPAS, we're applying ready-to-use formulations. So again, reimagining how those products are being applied and eliminating some of those challenges. And don't get me wrong, there's several products in the market that are fully compatible with most fertilizers, and that's a viable delivery mechanism, but not all of them are able to be applied in that manner or applied at the dose rate that you get the desired effect. So we're reimagining how you get those products to the furrow, to the seed, and trying to get to a more effective, more consistent return on investment. Because our experience is, at the end of the day, whether it's a synthetic or biological, the grower wants predictability and reliability. So if we can make something more reliable by delivering it in another means from traditional, that's what we're trying to do with the SIMPAS system.
That's great. Adam, I'm going to have you build on this a little bit from an agronomy perspective. Like, what are your thoughts on fitting in with grower practice to do better with these micro products?
Yeah, I just kind of had a side thought, you know, while Jim was talking, and you know, and when you consider agronomy and pairing an organism with a seed, right? They're both living. And some things that we've seen in testing in the last few years are some of the products, never mind biologicals, can be deleterious just to the seed that we're planting in proximity. So when Jim's talking about the SIMPAS system and being able to prescriptively dose if it's able to go in between seed or maybe a 2x2 in other cases, those types of things are going to be beneficial to the biological too. So I think as the industry and we're bringing these things to light and people are paying better attention, I think they're getting better value out of the products they're buying, because like I said, 1034 was mentioned and how there's varying qualities. And then on top of that, you have potential contamination at some of these large plants. So getting it away from the organism and getting it away from the seed, I think are beneficial for being creative with these application systems. If I were to design if someone were just to get into the retail space, or a grower wanted to apply their biological organisms, whether seed or in furrow or something, I think my answer would be pretty universal. Try to keep that organism away from everything else as long as possible. Even something as mundane as water, depending on your water source, could be deleterious with chlorine levels. You know, I'm lucky enough in our immediate area, you know, we don't have rural water, but not too far away in Southwest Minnesota, and probably lots of the people tuning in can utilize rural water and has chlorine. So there's remediation techniques that can happen there too. But I think that's where I would start, would be just keeping things away as long as possible. It would just deliver the most living organisms in the best fitness, because there's, you know, the tighter that you're delivering, the counts we're delivering, but it's also, are they, the ones that we're delivering, are they in good shape? And then maybe another comment would be, the organisms we're all dealing with are so different. You know, when we're just talking Brady rhizobia versus some of the other biofertility products in the marketplace, some are a little more fragile than others. It was once told to me, and I like to repeat it, you know, our bacillus products are kind of cockroaches. You can beat them over the head, treat them poor, and they'll survive. So understanding that, understanding the organisms, talking with the companies that you're buying these products from, and making sure that you're doing the steps to ensure the best practice.
There's so much we could talk about.
There is.
There is. We're almost halfway through, so I'm trying to be mindful. Let's move on to the impact of formulation, because I think, you know, all of you have had experience, I think, with liquid and dry, and maybe comment. I'll start with you, David. You know, at New Leaf, you have had both liquid and dry formulations. What has been your experience in how formulation impacts choices on delivery, performance of the product potentially being impacted?
Well, there's a place for both, for sure. I think the ideal place to be is have options that the grower can make fit into their application methodology. And sometimes that's dry, sometimes that liquid. So all things being equal in terms of the product's ability to perform in either formulation, I think there's a really good place for both. And row crop, in my experience, liquid was typically the way we were handling products, and growers were set up. They were either going through a sprayer unit or through a planter setup. In the specialty side of things, it tends to be more powder. Like I said, we're going into water. It's very convenient and easy to use. So the more we can formulate products to fit grower application, grower choice, I think the better.
Okay. And Dan, I'll just have you follow up on formulation and maybe some of the trends you're seeing or things that growers are telling you guys they want to see.
Well, I think David hit on a lot of it. It depends on the segment, and I agree fully that there's a place for dries and liquids. As you think about the infura market that we think about, the biggest thing you have to worry about really, well, there's several things, but as you get to the grower level and you pass off a product to a grower, the first thing you're absolutely not going to do is cause physical compatibility and clogging issues. If you do that, you've lost the ball game. So in some cases, when you're mixing in liquid carriers, physical compatibility limits the use of some of the dry products there, unless you can absolutely be convinced that you can throw something in a tank and you're not going to plug a tank. So that's a big one. And, you know, as you think about formulations in general, there's lots of things you got to think about. You know, first of all, you think about applicator safety. So you think about liquids versus dry, handle these things safely. So all of that comes into play as you develop these products. First of all, you got to have an efficacious product. You've got to be able to make it affordably so you can sell it. You got to make it safe so the handlers are safe when they handle it. And then you cannot cause physical mixing issues at the farmer level. So we take all of those things into consideration, Jay.
Yep. And Jim, I don't know, as you guys are developing your platform, is how formulation's impacting your decisions, if there's anything you want to comment on there.
So we've built a system to handle both granular products as well as liquid products. If I were to say, where do growers have preference, they tend to have more preference towards liquids. They'd rather pump than pour. With granular products, there's a segment of granular products out there we've had success looking at reformulating some dry powdered materials to avoid those kind of mixing challenges. You create a slurry and impregnate them on a granular substrate or carrier. So we've had some success doing that. We've had success with creating some ready-to-use liquids. But again, it just depends on the product you're dealing with. How stable is it? How many components are there? I'll give you an example. So we sell a product called Invigorate. We acquired when we purchased a greenhouse here 18 months ago, and that has 22 different materials in it. And again, the way we have employed to deal with those compatibility issues, they're co-fermented, right? So instead of fermenting individual components and then putting them together in a formulation, we co-ferment up to 22 different bacteria, microbes in the same fermenting system. So again, we've found a way on the front end to deal with that compatibility to avoid problems on the later days when you're trying to apply them in the field. So again, it just depends. Every one of them is unique, every one of them. And we've tried to develop a SIMPAS system that can handle lots of variable types of products. And with our granular experience, we have a segment that uses granules, and we have a segment that's very interested in mostly liquids. So our goal is to serve both needs.
Okay, great. We're going to move on to the next segment, which was more tailored around what we're delivering with. And I think each of you have already commented on the compatibility issues, and maybe we can delve a little bit more into that. Maybe more from the perspective of what are the limits of what we can deliver these living microbes with. Adam, you had started out by saying, ideally, we should just deliver them by themselves and keep everything else away. Maybe you can, we'll start with you and add on to what you were saying. What are our limits here?
Sure. Again, it goes back to what the organism can handle, right? And that's where a good compatibility and testing supporting the products is key. Yeah, in a perfect world, I guess I would want to build a system that's flexible. There's a lot of products that we carry, that a lot of people carry, that are pretty darn tough, and you can beat them up and they still perform. But I'm thinking down the road, what's the next product that's going to come out and provide value to the farm gate? So don't limit ourselves in just the easy button all the time. So keeping it separate, requesting, compatibility with the products that you're running, talking with representatives from the company. Of course, everybody's increasing their knowledge base a little bit. Maybe we'll find the good questions to ask when you have the opportunity to talk with your providers. Compatibility is it was touched a lot of ways. A grower's compatibility is exactly what Dan talked about. Is it gonna stop production? Is it gonna plug something? Compatibility to me is something completely different. Mine is, does it hurt the organism? Does it reduce its fitness? Does it compromise what it's designed to do? Like I said, in a perfect world, we're all dealing with different strains of bacillus and we don't have to have too much concern, but that's not reality. Shelf life, on seed stability, all these things. One thing I would like to add on seed, it's not only the delivery method of the seed, it's what happens after the fact. And in a lot of cases, we like to lubricate our seed with different talc type products. And I think it's pretty universal to state that if you're applying a liquid overtreat to your crop seed, there's a dry time that needs to go on naturally and prevent those cells from lysing, breaking, or drying too quickly. So if you are going to apply a seed lubricant like a talc product, make sure that seed has been treated and sat off to the side for a while to naturally dry down before you shock the system with a seed lubricant. That's going to greatly affect the performance and the longevity of the organism on the seed.
That's a great point. David, I don't know if you want to comment on in a greenhouse setting, because it is going into so much liquid. Have you seen any issues with limits in terms of nutrients or, pH or things that are trying to be kept or consistent in a greenhouse setting with microbes?
So a couple of things here based on what the guys are saying. I don't think synthetically there's a lot, at least at New Leaf, we could consider delivering our microbes to the grower with or in combination with. But where we do a lot of work in our R&D is, and there's a lot of companies emerging with prebiotic or non-living biological formulations that are plant beneficial, soil beneficial. Though a lot of those ingredients are powder and very low use rate. I think there's a lot of opportunity to think about how you combine those kind of components with live microbes to produce a more broad spectrum finished good. And in consortia products where you've got combinations of microbes that all get symbiotic with the plant a different way and affect plant metabolism in a different way, or the soil, I think, we do a lot of work in that area as well. So I think biologicals with biologicals is a huge opportunity. When we get to the producer, pH is probably our biggest concern. Low pH is death for our microbes. So we do have to be careful in that regard. And that's probably the number one thought when we're going into a liquid system. We're very conscious of what that system is from a pH standpoint.
Yeah, for sure. A liquid system different from a soil system where you do have a little bit more buffering capacity potentially, right?
Yeah.
All right. And Jim, to kind of round this discussion out before we move on to the next segment, if there's anything else as you're developing systems on the platform, on the SIMPAS platform, are there any limits that you guys are considering in terms of
there's
compatibility?
limits. So some of the comments that have already been made about not slowing the grower down. So with the advent of high-speed planters, you start getting into some physical requirements in terms of what's the dose rate that you're trying to apply. We've had some biological products that are targeting dose rates of 10, 15 or more pounds per acre. And when you're traveling at 10 miles an hour, is there a system capable of applying that much material at 10 miles an hour consistently? Or on the other end, you have some very, very low use rate products. And again, it gets back to if I'm applying, whether I'm in a carrier or not in a carrier, if I'm trying to apply a few ounces per acre, that's not a solid stream of material. So I have to create a solid stream of material through diluting or some type of ready-to-use carrier, or put it in a carrier stream and get a homogeneous blend so I can deliver a consistent dose rate across the area. So planter speed has become another factor in how do we formulate these products. And it's very grower dependent. I had a conversation with a grower up in New York State. He runs a commercial custom planting business. He runs 4 planters, and he told me his grower database has 102 different soil types across his business. So he's got to think about how these products work, how they don't work in all those different environments. And again, that's why we've tried to develop SIMPAS, that the more we understand about where something works or doesn't work, the comment about low pH is death to our microbe. Well, let's not apply in those areas where you have those conditions, right? Or let's focus the delivery of the material in those areas where you can get the most economic benefit. And so that's what we've tried to do with the SIMPAS system.
Yep, great segue. We're going to move into our last segment here, which deals with bringing together what I would say precision ag and precision delivery technologies with biologicals. So I think that's a great segue in terms of understanding through data, right, what we're delivering into and being able to hopefully prescribe better what we're delivering. So Jim, maybe we can start with you. From an equipment and data perspective, what trends are you seeing and how we're going to impact improving the use of biological products?
So traceability is a key discussion these days. So one of the things we've done with the SIMPAS system is we've created a readable, writable RFID chip that goes on every container. So we can track, so there's an automated as applied record that's created. So it's another data layer that's being created that grower can look at. The other thing we've done is we've built into the system a feedback loop. So we employ a company called, or a software package called Ag Veritas. So the grower is making prescriptive applications of a product within a management zone. We're including an untreated block within that management zone that represents about 2% of that management zone area. And then at the end of the year, he submits his as-applied records and his yield data to Ag Veritas, and they go and do a statistical analysis and provide him feedback in terms of what type of return he was getting for the invested input. Again, so that grower can then make decisions with his agronomist about, do I expand the use of these products? Do I retract the use of these products based on their performance within specific environments?
Now that's so exciting to hear things like this happening, because we're moving this, these are going to be step changes, right? That are going to keep moving us into this new stage of agriculture. So great stuff. I don't know who wants to take this on, this question on next, but maybe Dan or Adam, from an agronomy perspective, as we're bringing data, precision delivery, precision ag together, what trends are either of you seeing?
I'll kind of start, Jane. A good example for us, we're in the nitrogen fixing microbe business, and those microbes pretty well fix nitrogen across the entire landscape. But the value that they bring differs, like Jim said, by zone in the field. And where the precision piece for the nitrogen fixing microbe marries up with the farmer is really in the interface between those guys using variable rate nitrogen. So you may apply the microbe uniformly across the entire field, but then the value of that to the field, the variable rate nitrogen prescription may change based on the value of the microbe, say in a high nitrogen loss zone versus a low nitrogen loss zone. So we have some tools now and some analytics, like Jim was indicating, that will allow us to help those agronomists and those farmers efficaciously and economically use multiple biological products and also adjust their inputs like nitrogen. So I'm pretty excited of where the space can go.
Yeah, me too. Adam, anything you want to add there?
Well, Dan covered it well. I guess the only maybe little thing would be the year-to-year variation is the real, pie in the sky, right? A lot of our data analytic, it's kind of Monday morning quarterbacking. It's analyzing the game after you played it. So if we could figure out trends and maybe weather patterns or whatnot, I mean, that's what excites me, because I think the industry is doing a fantastic job collecting and generating all this data and even sifting through it. Now we're getting good at that. But that's really, like I say, that's the pie in the sky that I'm hopeful someone is able to crack that nut at some point.
It's okay to have pies in the sky.
Yeah, and the one thing that I'll build on what Jim said too, in a system like SimPass or variable rate controlled point injection systems where you could turn off a single input going across the field, that expands your analytical capability because you're not turning off a whole system. For example, if you're putting out our micro with 4 other inputs, you can theoretically just turn off any components you want, and then you have a check zone, so you don't have to turn everything off. So that's actually a really powerful tool if you think about it, and then you can really use precision ag analytics to really dial in where these things really pay and where they don't.
And I think we'll get there, right? I mean, we're moving in that direction anyhow. So with equipment, at least. David, did you have anything else you wanted to add from what you've seen? Any trends?
Well, that's great stuff. I would just say, we're starting to use precision ag as we take pipeline isolates to the field and try to understand performance. We're using precision ag so that as the field varies, the conditions vary, we can understand the performance changes as well, which can be significant. So yeah, this is a space that I think has got a lot of opportunity to it. We're working with AMVAC, their SimPass system. I'll echo what Dan said. That's the kind of technology that would allow us to commercially manage that variation. And I think that's really, really important.
Yep. Variation is key here, right? And understanding it. we're at the point where I think it's good to open up the live Q&A. I know we've had a couple of questions in the chat box, so I'm going to pull that up and we'll do our best to get through as many as we can. Great to see so many folks introducing themselves. Yeah, get to questions here. All right, I think this is a question here. Can we assume that optimal delivery can benefit from the best starting microbe if we think about nutrient efficiency? So if we're having, I think the point here is, are better microbes going to help us improve performance within this discussion on delivery? We can't have just bacillus being delivered in the field, right? So I don't know who wants to take that one. I know we are seeing better microbes.
I think that goes back to some of the innovations and I'll use 3-Bar as the example. So 3-Bar has developed a system that can deliver a more viable microbe to the field based on packaging, right? And we've seen other innovations in packaging, things as simple as with one of our biological products, we had to put a vent in the cap as it has a gassing effect. And again, you want to make sure that that grower retailer has a positive experience with handling that container. So everything from container innovation to learning about where these microbes work best and how do we deliver them through our supply chains in the most viable state. I think all of that goes into a more positive experience that grower can experience with those products.
Yep. I think an add-on to this as we finish up this question further, can the panel comment on how is industry discovery and screening of microbes today? And is there a better place to discover, screen, or modify a microbe as a starting point to know that it's going to do better in the field? Considering how we're doing discovery research today, is there maybe a better way that incorporates delivery more? I don't know if anyone wants to take that one.
I can speak to that. So we have a library of about 12,000. I mean, we collected that and we continue to collect. We have internal capability to DNA sequence those microbes and understand exactly what they are. And then we have what we call a prescriptive biologics platform that really helps us understand what the performance of that isolate should be. So yes, it's all, at least in our case, that has directed us in terms of what we research and what we look to, develop for commercial sales. So yeah, I think that's a really important question, and I think there's a fair amount of work being done in that space by a number of companies.
Yep, we haven't really talked about genetic engineering or genetic modification. Dan, you may want to take this one. How is that going to impact? We're seeing better microbes. Is that one innovation, one avenue that we can see them do better through delivery in the field?
I think I will just suffice it to say that the synthetic biology tools that we have access to today, compared to even as little as 10 or 15 years ago, are giving us the tools to build better microbes. There's no doubt. I mean, and certainly we leverage those tools at Pivot Bio to deliver, for example, our nitrogen fixing microbes that are going to work in an environment that is created by the farmers. They're not going to shut down in the presence of a synthetic fertilizer, for example. So, those tools are extremely important, and I think they're going to be extremely important to taking biologicals to the next round. We're going to build better bugs. There's no doubt about that across the industry. And David touched on it too. One of the big hurdles is, how do you know which one of these things to take to the field? When you got libraries of tens of thousands, and then when you go to the field, well, where do you start from even a rate or a combination standpoint? And then you have exponential combinations of things to test, and there's no physical way to test them in the field. So we've got to have those upstream data mining tools to tell us what to take to the field, and I think that technology alone is going to speed up discovery and development. So
sure,
lots of fun tools out there.
Yeah, and do you think that that's a space that that there needs to be innovation in, and being able to speed up that discovery process and figuring out what's going to better work in the field before you get it to the field? Maybe.
I would take anything that I could get in that space, to be honest with
All
you.
right.
So, but on the flip side of that, Jane, what you also find out is even the best lab and upstream greenhouse trials don't always predict what's going to happen in the field. So
That's right.
what happens in not just the field, but grower fields under real world conditions, it has to be really your last stage in the development process. And so we're not going to replace that anytime soon. We just got to be able to narrow down the number to a point where it's manageable in the field to trial.
Sure. All right. I want to thank James for those questions. The next question I think here deals with, we talked a good bit about compatibility today, but will liquid fertilizer pH and purity affect results? And this comes to us from Brett.
I think the answer is it depends on the microbe. Some yes, some no.
True.
So again, that variability we see with fertilizer, there's equal variability in how robust are the microbes and whether or not that will be a factor or not. So it's really microbe dependent, I would say.
For sure. And I think one of the things we worry about in thinking about compatibility is, we can test X, Y, and Z, but we don't know what the grower is really going to use, right? They could add things, they could combine them. And I don't know how to, I don't know if you all have any thoughts on how to get past that, because there's endless combinations out there, and maybe share kind of your approach to, you can't test everything, right, for compatibility. Are there any good approaches to handling this?
Well, I think there's also different objectives, right? So there are people that are using biological products to displace synthetic chemistry, and there are people using biological chemistry. I think the more dominant use, particularly foliar, is they're going to go in the tank with a synthetic, and you're looking to either mitigate resistance, you're looking to add another mode of action that's more complex to overcome resistance. You're using them in a different way. It's not an either or. It's one thing that I would say is a big gap in the industry is there aren't a lot of really good formulators out there that can take synthetic chemistry and biologicals and deliver the same kind of consistent shelf life that synthetics have enjoyed over the last few years. So that's a gap. And again, you have different objectives. What do you want that biological to do? Is it supplementing or is it replacing?
Good point. All right. We're down to a few minutes. I'm going to try to get to as many of these questions as I can. What understanding do we need of the soil microbiome to ensure these nutrient efficiency microbes are working? So kind of back to understand that data, right? What's already there? Have you guys had any experience in trying to use those types of indicators, or what are we missing there?
Well, we have a little bit of experience working with some of these startup companies that are looking at predictability of different pests. So there's soil DNA to tell you where you do or don't have different pests, so you can target those areas. We're working with some data mining companies. I'll give you an example. We were supplying the as applied data and the yield data. They had over 62 other data layers they could access from public databases. So then you start using AI to think and say, to get to that answer, where are these products performing at their peak? What's the environment that they work well or don't work well? And it's a lot of analytics. And I think we've only scratched the surface of where that could go in the future.
No, that's great. And I love that you brought up AI, because, data is going to be key. And we do have some great companies out there working on understanding what's there, but how do we start to map it with what we need to put there? I don't know, as we kind of close this out, probably a good question to ask is where is this headed? Are there areas where innovation needs to be happening, and any kind of final thoughts as we work to end the webinar? So I don't know who wants to start This will be kind of our maybe our last time around the horn. Innovation, closing thoughts around how we can start to bridge this gap.
I think the whole area of remote sensing, everything from electronic noses, drones, how do I get in front of the problem before it becomes a big problem? Whether that's satellite imagery, infrared imagery, is there a way to have more predictive, see the problem and treat the problem, or predict the problem and treat the problem? I think that's an area that will grow exponentially over the next several years on top of the innovations we've already spoke to.
Okay, great. No, love it.
Yeah, and I think another place, we didn't touch on it today, but the development of all these carbon markets and the ability to sequester carbon in soils. You've got companies that are now building technologies to intercept greenhouse gases from some of these agricultural crops. So, what Jim just said, I think is even important in that sector. And there's a lot of innovation that we need to focus just in that whole climate change space as it relates to agriculture. And that's just getting started. We're just scratching the surface there.
Yep, great points. Thanks, David. Dan or Adam? Any closing thoughts or areas of innovation that you're thinking about?
Well, I'll still say that I think the synthetic biology space is really going to continue to deliver dividends to the biological sector, and in a fast kind of turnaround time point that's a little different than your traditional GMO type approaches that take a lot of years and a lot of investment. So the synthetic biology front, I think, will continue to deliver. And I'm kind of excited about that space. All of the digital pieces that Jim and David touched on as well, they're the tools that we're going to use to be able to scale testing, scale product characterization, being able to deliver a product where it needs to be delivered for growers. Lots of fun things going on in that space. So I think the biological space will continue to thrive, and it's an exciting time in the biological world right now.
Totally agree. Adam, any parting thoughts here?
Innovation, to me, I think we're building it right now. All of the things we've talked about thus far are kind of in the now. We're testing the soil for now, but as more and more growers are mapping applications of fertilizer and tracking their crop. In the past, it was, what did you plant there last year? Was it corn or beans? That's kind of all we went backwards in time. But as we start to build those data layers over years, combined with yield maps and everything else, I think we're developing those layers that are going to help us in 5 years. We just have to keep building those layers. The cake's in the oven right now.
Yep, for sure. All right, and I think what's exciting to me is now we're seeing more and more growers, instead of being so skeptical of biological products, I think they're looking at them as being the tools of the future. So I think that's fair to say, and it's because of the work that I think as an industry we've been doing to show that these products can work, including how we're delivering them to the field. So I really appreciate each of your input today. We covered a lot of ground. There's a lot of challenges ahead and hurdles we have to continue to deliver, but appreciate each of your time. And of course, thanks to all of those of you that joined us live online. As mentioned at the beginning of the webinar, we'll have a link available to share the recording here shortly. So thank you to everybody and have a great day.
Thank you. Have a good day.
Yep.
Thank you.
Thanks.